Today we'll be hearing from three leaders in the field of prostate cancer research and clinical care. We have Dr. Kelly Parsons, Dr. Rana McKay, Dr. Christina Jamieson joining us today to talk about prostate cancer. They'll be sharing their very unique perspectives as both clinicians and researchers in this field. Here at the Cancer Center, we're very fortunate to have a really uniquely collaborative culture where physicians and research faculty work very closely together to accelerate advances in clinical care, in research, in really coming together to advance new treatments and understanding of the biology of many different cancers. So for our seminar today, we really want to thank the members of the Sanford Stem Cell Clinical Center, the Sanford Consortium for Regenerative Medicine, the Cancer Center, and the team here for bringing together these outstanding speakers. So first, we'll get on to introducing our first speaker. It's a pleasure to have Dr. Kelly Parsons here. He's a board-certified urologist who specializes in diagnosing and treating prostate cancer. He directs a variety of clinical trials for the Department of Urology at the Moores Cancer Center. He also leads the multidisciplinary prostate cancer clinic, which Dr. McKay and Dr. Jamieson, I believe, are also involved in. Focused on delivering advanced cancer care through a coordinated team approach, this clinic enables men diagnosed with aggressive prostate cancer, either early stage or metastatic, to see him, a medical oncologist and a radiation oncologist, on the same day. Dr. Parsons has published over 130 scientific research articles, edited 4 medical textbooks, received multiple research grants, and serves as an editor on 3 scientific journals. He's won numerous awards for his scientific research, lectures extensively across the US and in Europe and Asia, and serves on several national committees. Dr. Parsons completed his residency and fellowship training at Johns Hopkins School of Medicine in Baltimore. He received his medical degree from the University of Pennsylvania School of Medicine in Philadelphia and holds a Master of Health Science in Clinical Investigation from Johns Hopkins Bloomberg School of Public Health. He's really an expert in nerve-sparing robotic surgery, cryosurgery, and magnetic resonance imaging of the prostate, and is internationally recognized for his work in prostate disease. He's been repeatedly named a top doctor in San Diego Magazine's Physicians of Exceptional Excellence annual survey performed in collaboration with the San Diego County Medical Society. He's also been recognized as one of America's best doctors. So thank you, and please join me in welcoming Dr. Parsons.
Well, thank you very much. That was a very kind introduction. I'm Dr. Kelly Parsons. I'm at the Moores Cancer Center, and I'm a urologist, which means that I'm a surgeon, but I'm also the doctor who is most involved in actually detecting and diagnosing prostate cancer. And I think to that end, in addition to just citing my extraordinary colleagues who are part of the discussion today, Dr. Jamieson, Dr. McKay, I couldn't ask for just a more outstanding group of folks to work with. I'm just so delighted to come to work every day with them. And the agenda, I think, along the lines of what I do, my part in the team, because I am a urologist, because I am someone who's involved in detecting and diagnosing prostate cancer, I did want to take a little bit of a step back as an entryway into the conversation today. And first talk about prostate cancer in general. So what is prostate cancer all about? How is it diagnosed? Secondly, talk a little bit more about the team approach that was alluded to in the introduction. And finally, close with a introduction to cutting edge surgery. And I put surgery in quotations there because you'll see the significance of the quotation marks a little bit later. So first off, prostate cancer, a primer. I think one of the things that I am asked the most about as a urologist by medical students, by folks who are not in the medical field is what exactly is the prostate gland? Because we hear a lot about it, especially in older men, but we don't really know what it does. Well, the prostate is this very small gland. It sits right here. It sits underneath the bladder. It sits behind the pelvic bone, sits in front of the colon, the large intestine. And it's a gland that's involved with reproduction. It contributes to fertility. It produces most of the fluid that's involved in semen, and it's important for allowing sperm to live outside the body. I think I've covered myself here with the entire political spectrum from right to left. This is a selection of men who grappled with prostate cancer or who have grappled with prostate cancer over the course of their lives. And I think it points to exactly how common this disease is. And incidentally, the older folks in the audience will recognize that this is General Schwarzkopf, Stormin Norman, rather, as he was known. I had to actually add Ben Stiller, the actor, to this slide because I was giving this presentation to medical students year after year. And after a while, I reached a point where none of the medical students knew who General Schwarzkopf was. Ben Stiller, they're familiar with, and that's when the slide had to be brought up to date for the younger generations. But the lifetime risk of diagnosis is 1 in 6. For the folks in the audience who've been touched by prostate cancer or who know folks who've been touched by prostate cancer, you can see how incredibly common it is. Well, what causes prostate cancer? Well, first off, one thing that we know about prostate cancer is that it feeds on the male hormone testosterone. And this is just a schematic that simply shows that when you have these signaling pathways from the brain starting up here, going down this way toward the prostate cancer cell, which is represented here. All of these pathways, all of these sorts of things result in the prostate cancer feeding on the male hormone testosterone. And when it feeds on testosterone, it grows. So if we take, in fact, testosterone away, which is what we do in certain situations in folks who have advanced cancers, the prostate cancer will shrink and die. What about other things that we know are associated with an increased risk of prostate cancer? Well, first, genetics, certain genes that folks are born with, family history that can contribute to it. Secondly, age. So as folks get older, the risk of prostate cancer goes up. So a 20-year-old man is not generally going to be diagnosed with prostate cancer, but an 80-year-old man might. Finally, African ancestry. For reasons that are not entirely understood, African-American gentlemen are at increased risk of being diagnosed with prostate cancer. Importantly, if they are diagnosed and appropriately treated, they have the same exact rates of cure as gentlemen of other races and ethnicities. What other kinds of things are at play? Well, obesity, red meats, fatty foods, all of the things that taste really good that are associated with heart disease, all of these things also seem to be linked to an increased risk of prostate cancer. Well, how is it diagnosed? I think anyone, certainly all of the men and their families are aware who've been diagnosed that it's usually diagnosed with a blood test, what's called a PSA blood test, and then also a finger examination of the prostate. We call it a digital rectal examination. I think this is instructive because a lot of men don't really understand when we do a physical examination, why are we doing it? What are we feeling? We're feeling the prostate right here. And nature has cut us a break in that about 90% of the prostate cancers actually occur on the backside of the prostate where it can be detected through a finger examination. And so men who are interested in being screened for prostate cancer should keep in mind that we do recommend both the blood test and the finger examination, generally on an annual basis. Well, at UC San Diego, as we alluded to, we do believe in a team approach for treating prostate cancer, and we are dedicated to that team approach. And we think it's very unique in terms of what we're able to offer, certainly to the community of San Diego, and it's in line with many of the other big universities across the country. The analogy that I like to use here is Michael Phelps. Michael Phelps, as you know, is one of the most successful Olympic athletes of all time. He won 8 gold medals in a single Olympics, and people tend to associate the sport of swimming, in fact, with Michael Phelps. What a lot of folks don't know is that nearly half of the medals that Michael Phelps has won, including all of the gold medals that he won in Beijing, he won on relay teams, which means that he wasn't winning just alone by himself, swimming by himself. He was winning because he was swimming with 3 other athletes. And so I hold that up as what we strive for every day. Sure, everybody would like to be Michael Phelps individually, but what we believe at the UC San Diego Cancer Center is that truly to be Michael Phelps, we need to work as a team. And that's how we get better every single day. And that's how we put our patients first and treat our patients first every single day by working as a team. Well, operationally, what does that mean? Well, there are 3 general arms in terms of treating prostate cancer. The first is what our oncologists, Dr. McKay, use, which is medications, typically. The most common medication, at least that folks I think are familiar with, they always think of chemotherapy. We don't use chemotherapy very often in prostate cancer, but there are other kinds of medications that we use in prostate cancer. So that would be the first arm. The second arm is what I do for a living, which is surgery. And the third arm is radiation. We don't have any of our radiation oncology colleagues on the call today, but we work with the best, and they are an integral part of our team. And all of those together, those 3 arms together, those 3 elements together, blend to achieve the best chances of a cure, complete eradication of the prostate cancer. And so after much planning, we achieved a point where we were ready to open our clinic, which we did on January 24th, 2020. This is our entire team, myself, Dr. McKay, Dr. Rose, our outstanding nursing colleagues, and that was the first day we started seeing patients. And I'm proud to say that we've since been able to expand our services. We have worked through the pandemic. We realize that while keeping our patients safe is absolutely first and foremost, always our first priority, we also want to be able to bring cancer care in a safe way to the patients who need it the most, the cancer patients. And so we have been able to continue the clinic even through the pandemic, and occasionally what we'll be doing is even doing it through telehealth, through video visits. But these are some of the components, the advantages of what we believe the cancer clinic is all about. Well, at the center, it's all about better care and us trying to make care better for our patients every single day. And some of the elements that go into that, some of the advantages of having a multidisciplinary clinic are comprehensive attention in one setting, in an 1.5 hour long visit, a visit, in fact, that's one visit with 3 of us, 3 separate doctors all seeing folks at once, and importantly, one treatment plan. So we come up with one treatment plan while folks are there, the 3 of us. I always like to tell folks, our patients, 3 heads are always better than 1 in terms of coming up with prostate cancer treatments. And finally, and we've heard this a lot from our patients, improved communication. Because all 3 of us are there, all of our teams are there at once, and we're able to communicate things, I think, a lot more effectively and a lot more clearly. Well, finally, cutting edge surgery. I put surgery in quotations simply because it really isn't necessarily surgery. This is something that I'm very excited about, something that we've greatly expanded at UCSD over the last 2 years in particular. This is former President Obama. He's sitting at a robotic surgery system. And this was at the Cleveland Clinic, actually. And this is what we use at UCSD. It's the most common form of what we call robotic surgery, using a robot to take out the prostate. Over here on the right, highly precise instruments that are directed depending on the surgeon's hands. Very, very small instrumentation, very precise surgery, very safe, very effective. But to take it a step further, what if we didn't even really do surgery? What if we left the prostate where it was and we somehow treated the tumor without removing the entire prostate? And that's what we've been striving to do at UC San Diego. The approach we favor is something called prostate cryoablation, which involves freezing the cancer and the prostate around it. It's Medicare approved. Okay, so meaning that it's not experimental. It's not something that patients have to pay for, certainly out of pocket. And it can be used in terms of initial therapy, so the first therapy that a patient gets. It can be used as therapy if someone's had radiation previously, now the cancer is coming back. Important to note, though, that it's not for every single patient. The basic concept is that if this is the prostate here that I'm circling around, and this is a high-tech ultrasound probe that we use, we take very small needles, we put them directly through the skin into the prostate, and we freeze the prostate with supercooled gases. And what we're able to do on top of that is to also use MRI imaging. So this is an MRI image, and this orange color here, that's where the tumor is. And we can use the MRI images in the operating room to actually guide ourselves to the tumor like a GPS, limit the area that we're treating, and therefore limit the potential for side effects. And really, thank you so much for joining us today. I'm just, like I said at the beginning of the talk, I'm just so excited to come to work here every day to work with my partners. Thank you so much.
Thank you, Dr. Parsons, for that fantastic overview of prostate cancer and what the therapeutic options are. Really phenomenal to see the collaborative work going on at the Cancer Center. And I'll move on now to our second speaker, Dr. Rana McKay, who's a board-certified medical oncologist who specializes in treating people with urogenital cancers, including bladder, kidney, prostate, and testicular cancer. Dr. McKay leads a multidisciplinary prostate cancer clinic together with Dr. Parsons, focused on delivering advanced cancer care through a coordinated team approach. This clinic enables men diagnosed with aggressive prostate cancer to see her and the team of doctors that Dr. Parsons mentioned at the Moores Cancer Center. As an associate professor in the Department of Medicine, Dr. McKay instructs medical students, residents, and fellows at the UC San Diego School of Medicine. Her research interests include the design and implementation of clinical trials to including novel biomarkers and therapeutic outcomes for patients with genitourinary malignancies. She serves as principal investigator on several early phase trials in kidney and prostate cancer. And as a clinical investigator, she's committed to advancements in cancer care that will improve the lives of cancer patients. Before joining UC San Diego Health, Dr. McKay was a medical oncologist at the Dana-Farber Brigham and Women's Cancer Center in Boston and an assistant professor at Harvard Medical School. Dr. McKay completed a fellowship in oncology hematology at the Dana-Farber Cancer Institute, Harvard Medical School. She completed a residency in internal medicine at Johns Hopkins Hospital in Baltimore, and Dr. McKay earned her medical degree at the University of Florida College of Medicine in Gainesville, Florida. She's board certified in internal medicine and medical oncology. And she works with the team in the multidisciplinary clinic. And we're really honored to have her here today to share her perspective as a clinician in prostate cancer. So I'll turn it over to you, Dr. McKay. Thank you.
Thank you so much, Leslie. That was wonderful, too kind of you. So we're going to dive right in and we'll switch gears a little bit, focusing on advancing the clinical application of genomics in prostate cancer, and just really some of the exciting, science that's been coming about, really diving into what are the drivers of this disease and how can we better target those drivers. You are well familiar with this data. Prostate cancer is the most common cancer in men in the United States, with its incidence peaking really in the early 90s with the introduction of PSA testing. And you can sort of see what's happened over time with regards to incidence in PSA. But our patients are actually living longer, and mortality is improving with regards to prostate cancer, with declining number of deaths. And that's largely been due to improved detection strategies and better treatments to treat this disease, which has certainly been exciting, but we're still not there yet. We still have a lot more to go to make sure that our patients are living longer, living better, and we can relinquish this disease from its existence. Dr. Parsons alluded to this already. The backbone of systemic therapy for patients with advanced prostate cancer and even localized disease has really been hormonal therapy that works to decrease testosterone levels or block the action of testosterone. And this work was conducted by Huggins and Hodges back in the 1940s, actually led to the Nobel Prize being awarded to them for the discovery that prostate cancer was stimulated by testosterone, and that taking away testosterone actually improved outcomes for patients. And then, nearly 75 years later, this is where the field is at. Lots of new agents entering into the treatment landscape, novel hormonal therapies, which have really populated how we treat the disease over the last 10 years, newer chemotherapies, radium-223, a liquid radiopharmaceutical, Provenge or Sipuleucel T, which is actually the first vaccine therapy to be approved for any solid tumor malignancy. Go figure, it was in prostate cancer. And then most recently this year, I think breakthrough discoveries with the approvals of olaparib and rucaparib for men with advanced disease. And I really think this is opening up a huge box with regards to genomic sequencing and the potential that that has for patients, because really now the introduction of olaparib and rucaparib has cemented in the ground that all men with advanced prostate cancer should undergo genomic profiling. And as we do more genomic profiling, we're going to understand what are the drivers of disease, resistance, and how can we better combat those. And so, prostate cancer is treated with multimodality therapy. These are the systemic treatments, but also surgery, radiation, as you can see here. And so what are the mechanisms that drive resistance for patients with advanced disease? And I like to break those down into what are strategies that are dependent on the androgen axis and signaling of testosterone, and the androgen receptor, which is critically important to the spread of prostate cancer and the what we call pathogenesis of prostate cancer, and versus what are AR independent mechanisms of resistance. And so you can see that delineated here on this schema. Prostate cancers can mutate where they can have more androgen receptor. So even a little bit of testosterone that's around can activate the androgen receptor. They can mutate so that new types of agents can turn on the receptor. So instead of testosterone, maybe progesterone or other hormonal agents can turn on the receptor. There can be other pathways that are activated outside of the androgen receptor. There could be pumps that actually pump out drugs out of the cell, so you can't get the drug into the cell effectively. There can be ways that the cancer cell can figure out how to not die when you are giving it hormonal therapy or giving it chemotherapy. There can be development of growth of blood vessels around the cancer cells that help it spread. So there's all these very interesting, elaborate mechanisms of resistance to prostate cancer and the therapies that we give a prostate cancer to help stop the spread of disease. And I think trying to understand how we can target each of these pathways is critically important to making our patients live longer. So there was a landmark study that was actually published in Cell in 2015. This was a huge collaborative effort, in part funded by the Prostate Cancer Foundation and Stand Up to Cancer, putting together large collaborative teams of scientists to try to better understand the genomic profiling in prostate cancer. And prostate cancer has sort of been a little bit behind the 8 ball with regards to other tumors, and it's given the sheer nature of where the cancer spreads to. The cancer largely spreads to the bone, and that has been historically very problematic with regards to actually being able to biopsy a bone specimen, understand the genomic makeup of that disease, so we can figure out how to better target it. But this is academic science at its best. Scientists getting together, pathologists, radiation oncologists, surgeons, all coming together to really understand, well, how can we do this? How can we get it so that we can better understand what is driving the prostate cancer to grow? And so there was this large effort to sequence about 150 men who had metastatic prostate cancer and actually sample their metastases, sample their mets, and try to understand what is causing these cancers to grow, or what is causing these cancers to be responsive to any given therapy. And from that work has led to the understanding that there are various mutations in advanced prostate cancer that drive prostate cancer growth, and probably about 80% of prostate cancers actually harbor some sort of mutation that is potentially targetable by maybe not an FDA approved drug, or not a prostate cancer specific drug, but a drug that is on the market somewhere for some other disease. or being developed. And one of the important revelations from this work has actually been better understanding the DNA repair pathway. So from this large series, we learned that about 20% to 30% of patients who have advanced prostate cancer actually have mutations in the way that that cancer repairs its DNA. And why that's important is because there are certain drugs that can be used to combat this exact molecular genotype. Additionally, what we learned is that 1 in 10 men who have advanced prostate cancer actually were born with something to predispose them to prostate cancer. They had a hereditary syndrome inherited down through generations that predisposed them for the disease. So this is a landmark paper published by Pritchard and colleagues in 2016, where they literally took up to 700 men with metastatic prostate cancer and isolated germline DNA from those individuals, meaning, just normal DNA in these patients, normal cells, not in their tumor cells, what they were born with, and actually showed that 12% of these patients actually were born with something to predispose them for disease. So why is that important? It's one, important because there are drugs that we can use to block these alterations. And two, it's important for cascade testing for men. And what I mean by cascade testing is that um, this could be something that could be very important for children, brothers, sisters. It could predispose the daughters of men who have prostate cancer to breast cancer and other diseases. And so it's critically important that we are doing the testing so we can figure this out. Without doing the testing, we'll never know. And so, 1 in 10 men, again, have germline alterations that predispose them to prostate cancer, and probably 1 in 20 to 30 men with advanced disease have a DNA repair alteration that could potentially sensitize them to a drug such as a PARP inhibitor. So just this year, breakthrough FDA approvals for both olaparib and rucaparib for patients who have advanced prostate cancer with DNA repair alterations. And, this is groundbreaking because this is the first time in prostate cancer that we have a drug or two drugs that are approved with a molecular indication, meaning if you fit into this molecular phenotype, you're going to potentially be exquisitely sensitive to this drug. And I think we are going to be seeing more of this in prostate cancer. And this, in essence, is what we call precision medicine. This is the power of precision medicine in prostate cancer, where instead of just giving one drug to everybody without any discrimination based on what is the underlying genomic makeup of that cancer, we're trying to personalize therapy and individualize therapy based on that patient's cancer's genetic makeup, where we give the right drug to the right patient at the right time. And by doing that, we hopefully improve efficacy, we decrease toxicity, and we're more prudent in our therapy selection for patients with advanced disease. So this is sort of where the field is going. It's been very exciting to be a part of this scientific endeavor, which really at the end of the day is targeted at, again, making patients live longer, making patients live better. And how UCSD has contributed to this story is we've designed a series of clinical trials that are focused on better understanding drivers of disease resistance, and trying to put therapies together that make scientific sense so we can improve the effect of any one given therapy, trying to better personalize therapy for men with prostate cancer. So I highlight here the results of a large phase 2 study of cediranib and olaparib that we recently conducted. This trial is fully accrued. This study was designed to actually figure out how to enhance the efficacy of the drug called olaparib, which is a PARP inhibitor. And this data will actually be presented at the GU ASCO symposium this coming February. We actually got invited to present an oral abstract presentation. I couldn't help but put in this curve demonstrating improved progression-free survival, meaning delaying the time that somebody would progress when they receive this combination compared to olaparib alone. And so how can we improve the lifespan of these drugs, make them work longer, work better in our patients? And cediranib is a type of VEGF inhibitor. And VEGF has a long story in prostate cancer, but there seems to be some synergistic effect with olaparib. I highlight here the results are a study that's currently ongoing of radium-223 plus olaparib. Radium-223 is a liquid radiopharmaceutical that actually delivers a small bit of radiation to bone metastases. And olaparib and radiation actually causes DNA damage, and olaparib actually impairs DNA repair. So we think that the two drugs in combination can actually result in improved efficacy. So we just finished the phase 1 portion of this study. We're about to launch the phase 2 portion, hopefully at the start of next year. This is a national study being led through the NCI, which is the National Cancer Institute. So we're very excited about being able to come up with combinations that potentially are rationally designed to improve the efficacy of these drugs. So that's in the metastatic space, and this is kind of our pipeline around PARP inhibition. I do want to highlight the study that is about to launch now. This is the phase 2 Neptune trial. And as I stated earlier, patients with metastatic prostate cancer, 1 in 10 of those individuals are at risk of a germline alteration, maybe something that they are born with that predisposes them to prostate cancer that could sensitize them to olaparib. So taking that same concept, we said, well, why are we waiting until they get metastatic disease before we give them this drug? Why don't we give them this drug when they're still localized so they never get metastatic disease and we can cure them earlier? So we've designed this trial called the Neptune trial, where we give patients upfront olaparib with hormonal therapy for 6 months, patients who have a germline alteration in their DNA repair pathway. So we suspect they would be very sensitive to this therapy. Do this and then remove their prostate and see what happens. The primary endpoint is pathologic response. This is a small study. It's just 32 patients with 30 evaluable. It's being run through HCRN, which is a national group that allows us to do this across multiple institutions. So we're hoping that this will open up at UCLA, Michigan, and Johns Hopkins, in addition to our institution. There was a similarly designed study in breast cancer, and this in a similar patient population. I know breast cancer and prostate cancer are not the same thing, but that demonstrated a pathologic complete response, meaning you look at the breast specimen after they got surgery and see how much cancer is left behind, a 50% pathologic complete response rate. So we believe that actually by doing this strategy up front, we can potentially cure more patients with prostate cancer. This is still early on. This is sort of a pilot proof of concept that yes, we can do this, but I think these are the kind of questions we're asking at UCSD. Segueing with that, the most important thing actually, predicated on this trial is we need to actually find who these individuals are that have these alterations. So in the localized setting, the prevalence rate of these alterations is probably 1 in 15 men, or 1 in 20 men. But for that 1 in 15 or that 1 in 20, it's huge to find that alteration. So what we need to do to actually be successful for a trial like this, and what's actually been recommended by the NCCN guidelines, is actually every man that has high-risk prostate cancer that's localized should be offered germline testing. And it's something that we're not doing very well, that we need to do better. So we've actually created an educational initiative within the clinic. So every man that comes into our clinic that meets criteria for NCCN guideline germline testing is offered testing. So we have like an educational one-on-one with a medical healthcare provider, and a patient can then decide, yes, they want to do the testing, or no, they don't want to do the testing. The goal is not that we get the test. The goal is that we educate patients, that patients make an educated decision, and they can decide whether they want the test. But every man should be approached. And I think this is critically important for those minority patients, African Americans, Hispanics, and making sure that this educational tool can suit the needs of all of our men with prostate cancer. So other really exciting things that are happening, we are part of the Prostate Cancer Foundation N of 1 working group. We just recently published kind of a white paper in Nature Cancer about accelerating precision medicine for people with advanced prostate cancer. How can we learn from our exceptional responders and exceptional non-responders to therapy? How can we better personalize therapy? So in this paper, we actually lay out the challenges and solutions for how we think we can accelerate precision medicine in prostate cancer, and the steps that are needed to get us there. And we're working with the NCI on a natural history study where we actually do just this, trying to understand who the exceptional responders are, who the exceptional non-responders, to better personalize therapy. Dovetailing with those efforts, we've actually developed the PROMISE Consortium. Literally myself and a couple of friends across the country were like, we need to be able to aggregate our data and better understand what are the implications of some of the molecular findings that we're getting back from our commercial tests that we get done, and how we can better learn from our patients, and how we can understand how any one patient is responding to any given therapy. So we've actually generated a national registry. Right now, this literally launched in the midst of COVID over the summer, and right now we're up to over 700 patients. We've got 19 institutions that are involved, and UCSD is on the steering committee, and we are leading these efforts right here at UCSD. On the heels of all of this, ultimately, yes, we need to learn more, but we need to get better drugs into patients in a streamlined kind of way. So working with collaborators like Misha Beltran, Chuck Ryan, and the NCI Alliance team, we've actually put forward one of the first basket umbrella studies in prostate cancer that is actually looking at trying to personalize therapy for men with advanced disease. So this was a trial that we presented at the Alliance. Just about 2 weeks ago, we presented it at the Prostate Cancer Task Force, where based on a patient's molecular alteration, they would be assigned to a drug arm that we would suspect would be associated with a response to treatment based on whatever biomarker they have. And so we think a trial like this is really going to be revolutionary for the field. And while right now this trial only has 5 arms, as we learn more and more drugs become available, we think we can expand this. We can add additional arms, and we can have the trial, we can learn from the trial and learn from our patients, but also work to better personalize therapy. So, my hope is that one day a trial like this will become a reality for patients with prostate cancer. And one day we won't be doing a trial like this for people with metastatic disease, but for people with localized disease, so they never become metastatic. But that's where the field, I think, is going, but we need to help get us there. And then this is another trial that is currently in the works. It's actually pairing an immunotherapy with an angiogenic agent. There's been some very promising data around the combination of atezolizumab, which is an immunotherapy, plus cabozantinib, demonstrating response rates on the order of 30% for men with advanced disease. We think nivolumab is a better drug than atezolizumab, and so we're doing a study with this drug combination. This is run through the Prostate Cancer Clinical Trials Consortium, funded through BMS and AstraZeneca. But the correlative work, we are still working on building out the funding program for that. And then Chris Jamieson, partner in crime in the lab, she's amazing, great researcher, is we've had a lot of interest in the Wnt pathway, and I'm just putting this out as a teaser for a phase 2 study of seramtuzumab plus docetaxel. And seramtuzumab is pretty unique. It was actually developed at UCSD by our own scientists and own investigators, and it actually targets cancer stemness and targets the cancer stem cell itself. And so we are pairing the combination of seramtuzumab with docetaxel in this heavy biomarker intense trial, so we can better understand, who are the right patients for this kind of therapy, who's best going to respond to a therapy like this. This trial is, through basically putting together various resources from every angle that you can possibly imagine to get this trial to launch. It's IRB approved, and we're working on actually activating it for our patients. So this is sort of up and coming. And, our translational research goals, aside from the clinical trials, we really want to be able to build a systematic platform where we collect clinical data, we collect, specimens, we're able to annotate the specimens with clinical data, so we can better learn about what's driving these cancers to grow and help overcome that resistance. And so this is sort of our infrastructure pipeline for translational work within the clinic. Identify patients. Patients may have been approached to participate in a lot of these translational efforts. But really, at the end of the day, the intent is to, link clinical data to molecular data, and not just molecular data, could even be immune data, so we can better target therapies for men who have advanced prostate cancer. So kind of in conclusion, comprehensive molecular interrogation of patients with prostate cancer will continue to refine our understanding of the disease pathogenesis. And really what's important is expanding the platform for precision medicine. The goal is to demonstrate the clinical utility of next generation sequencing and enabling widespread adoption in the treatment of men with advanced prostate cancer. We have an amazing team that's here at UCSD, and we also work with leading collaborators across key institutions, also work with industry. It takes a village. It takes more than a village to get this work done, but really this is where the field is going. And so excited to partner with you and partner with everybody here, Dr. Parsons, Dr. Jamieson, and others, to really get us to the next step, get us to the next level. So thank you.
Thank you, Dr. McKay, for that amazing tour de force of the clinical trials that are going on in this field. I mean, really just fantastic work going on here and across the country. I mean, really exciting to hear about your national registry and how that might be able to share advances across the nation. So very exciting things going on. With that, I'll move along to introduce Dr. Jamieson. Dr. Christina Jamieson received her BSc honors from the University of British Columbia in Vancouver, Canada, and a PhD in molecular immunology with Dr. Ranjan Sen at Brandeis University in Massachusetts. She showed for the first time that the T-cell receptor activates the pathway NF-kappa B. Dr. Jamieson did postdoctoral training at UC San Francisco with Dr. Dan Littman and then Dr. Keith Yamamoto, where she showed that TCR signaling crosstalk alters the transcriptional function and output of the steroid hormone receptor GR, thus inhibiting apoptosis or cell death. So she's done really amazing work in genomics and molecular biology in cancer. And she then joined the University of California, Los Angeles as an assistant professor in the departments of urology and human genetics, where she initiated her work on bone metastatic prostate cancer and resistance to androgen receptor or AR targeted therapy. Dr. Jamieson then moved to the University of California, San Diego to join the Department of Surgery and Urology, where she established new patient-derived xenograft models of bone metastatic prostate cancer, as well as a biorepository of surgical prostate cancer bone metastasis specimens. Dr. Jamieson is the first to show that the bone niche is sufficient to support castration-resistant growth of prostate cancer. She identified gene networks associated with prostate cancer growth in the bone niche and established a co-culture model system demonstrating that bone marrow stromal cells supported the castrate-resistant growth of prostate cancer cells. These findings supported the hypothesis that signaling pathways within the bone marrow microenvironment actually contribute to therapy-resistant growth of cancers. Dr. Jamieson is also using new models of bone metastatic prostate cancer to develop molecularly targeted therapies and immunotherapies. So we're really honored to have her here to talk about the bench side of this field and how that integrates with the work on the clinical side that Dr. McKay and Dr. Parsons are doing. So thank you so much, Dr. Jamieson, and I'll turn it over you to share your work with us.
Great, thank you so much, Leslie. I miss seeing you. We're actually neighbors. Our labs are right next to each other, and we've known each other for a long time. And so I miss seeing you because of all this COVID restricted activity. So it's really nice to see you. And thank you so much for the wonderful introduction. And, I've had a tremendous experience with my two colleagues, Dr. Kelly Parsons and Dr. Rana McKay. And so today I'm going to share with you a couple of the research stories that have developed at UCSD that I've been fortunate to be part of, where we've taken work that was products developed here, tested them in our models, which we've developed using patient material, which are then much more predictive of the responses. So let me just share my screen. So this was work that could not have been done anywhere else, only at UCSD. So I'm an associate professor here in the Department of Urology. I'm a PhD scientist, and I'm lucky to work with a fantastic team of urologists, and GU oncologists, as well as orthopedic surgeons, and then an amazing powerhouse of scientists here at UCSD. And so just for background on, my main focus has been on bone metastatic prostate cancer. And as many of you know already, prostate cancer unfortunately metastasizes. When it metastasizes, it typically goes to the bone, where it can cause a number of damages, pathologic damage, such as intractable bone pain, pathologic fractures, and spinal compression. There's currently no cure, but we're working hard on that. So I wanted to really address this aspect of prostate cancer in my research. And so I've worked to establish models using patient bone metastases, and I worked very closely with Dr. Anna Kalugian, who is in the La Jolla area, and she does the repair surgery. So when the prostate cancer is growing in the bone, such as here in the femur, the right hip, sometimes the growth will cause a fracture, and at that point, Dr. Kalugian comes in to do the repair, and she'll put in, a half hip replacement, and at this time removes a lot of the material, as much of the tumor material as she can. This isn't a cure, but it does help the patient tremendously. It reduces the pain, but most importantly, the patient is still ambulatory. But at this point, this material is then given over to me and donated, and we're so grateful to the patients who do this, donated to us. It's not considered anything, they don't keep it for any other reason, so it's incredibly valuable for our research, and we take it back to the lab and immediately use the living tumor cells to put them into mouse models. We put the human cells into mice that don't have an immune system, so that the tumors, if they'll grow, they'll grow, and the mouse immune system won't reject them. We study the tumors themselves using various genomic transcriptomics. We've looked at the tumor growth in the bone and the mouse bone, and it replicates many of the features that we see in the patients. And we've set up mini tumors in culture, also known as organoids, which you may have heard of. So with this fantastic collaboration with our colleagues, we've been able to collect so far 25 of these large amounts of patient bone metastases, and we've established some serially transplantable models, which means you can take the tumor and grow it in mice and be able to expand and have a renewable source of these tumor cells, which is incredibly important. And now we've also set them up in organoid cultures, so that we can now set up organoid cultures from all the patient samples. It takes, it's harder to grow them in the mice, but now we can grow them in culture, bone metastases as well as the primary prostate cancer. So our strategy then has been to take the bone metastases, implant them into the mouse bone, and then study their growth either in the bone or in a non-bone environment, and compare the effectiveness of different therapies. So what we found is that the bone is a much more resistant environment. And this is to show that a lot of the genomic rearrangements that occur in the patient are also maintained. So for example, here in the xenograft, xeno meaning foreign, graft into the mice, so human tumors growing in mice. One of the first things that we did was then to look at, well, what genes are active in the bone environment in the patient and in these xenograft tumors. And what jumped out immediately was this gene called Wnt5a. Now, this makes a protein that binds to a receptor, which by happenstance, the lab right next to me had developed a therapy for. So by just being in the right environment at the right time, we were able to see that this ligand gene called Wnt5a, I know it's a funny name, Wnt5a, which had been bubbled up and was kind of becoming very well known in prostate cancer, because it was found to be involved in bone metastatic prostate cancer that had become resistant to the androgen deprivation therapy. And so it turns out to be an important gene in somehow, giving that ability of the prostate cancer to grow in the bone and be resistant. And so because we saw it in our patients, in our models, and it had been discovered about the same time in the field as being a really important marker of this aggressive disease, we decided to go ahead and put our effort into testing this therapeutic called simtuzumab. And so what happens is, here's Wnt5a, and it comes from another cell, and it can then interact on the cell surface with these receptors called ROR1 and ROR2 ROR. And this antibody therapeutic had been developed against ROR1 for leukemia. And so it turns out that ROR1 is normally expressed in the fetus, but it can be upregulated, so the protein suddenly comes on in a number of cancers. First, it was discovered by Dr. Tom Kipps and the groups here at UC San Diego to be upregulated in chronic lymphocytic leukemia. And then as a group with Katrina Jamieson, my sister, and Dennis Carson and Charles Prusak, they developed this phenomenally specific and non-toxic simtuzumab antibody therapeutic, which they've already tested in phase 1 clinical trials and shown some efficacy for chronic lymphocytic leukemia. And so now this has also been looked at in solid tumors, and it was found that some solid tumors can also upregulate or increase the amount of this fetal or kind of stem cell, cancer stem cell protein. And so now there's additional clinical trials for CLL, and then at UCSD, and a trial now started in metastatic breast cancer. And so from these, incredible advances, and working with Dr. Parsons and Dr. Rana McKay, you can see the energy that we have for this. And so we have this unique ability and to take this very well-tolerated antibody therapeutic and apply it to prostate cancer, where its ligand, Wnt5a, has been shown to be a key signal that the cancer growing in the bone has now reached an even more aggressive state. And so we've now tested the simtuzumab in very early stages. We did characterization of expression levels and all this kind of stuff to really identify what's the best place where we're going to find responsive cells. And so we did a trial to mimic the clinical trial that Dr. McKay was telling you about, where we're going to treat with simtuzumab and do a lead-in and then add in docetaxel and have both. So the trial here in the preclinical trial was to grow our ROR1 positive model, patient-derived model from a bone metastatic prostate cancer. And we treat half the mice with a control antibody and then half the mice with simtuzumab for 2 weeks, and then start adding in docetaxel. And this is early results, but it's very promising. And what we see is that in this gray line and the blue line, there is a separation, and then in the tumor growth, which this was actually very large tumors, so this is pretty phenomenal to see a difference. And now when you start adding in the docetaxel, we were actually suppressed and arrested the growth compared to the control treatment. So now we're going to repeat the experiment, refine it, the dosing, and then in parallel, we're ready to go in patients. So this antibody is very non-toxic in all the patients that have been, most of the patients that have been tested so far for other cancers. And now we know that in combination with docetaxel, there's some promising results if we are looking at tumors that express the protein. Another really great, so I'm telling you like the latest data that we have in the lab, because it's so exciting. Another product that's been developed by the same group is to use this wonderful antibody against ROR1 and put it onto T cells. And this is known as a combined antigen receptor T cell. Now, T cells are like the super killers of the immune system. And so if you can load them up with an antibody that can then recognize the protein on a tumor, this is one of the most potent ways to truly eradicate a tumor. And so now we've got these CAR T cells that have the Simtuzumab antibody on the surface, and we introduced those cells into mice that had tumors that are prostate cancer tumors that express ROR1. And if you see here, the gray bars are the tumor sizes increasing in mice that only got control T cells, no antibody on the T cell. And then the blue bars, you can actually see the tumors start to shrink in mice that got the CAR T cells intravenously at an early time point. So this, again, is very, very promising. This is really preliminary, but we're very excited about it. And I think this is going to be a tremendous treatment for a very aggressive form of bone metastatic neuroendocrine type of prostate cancer. So we're very excited about this. I'll tell you very briefly, this is another product that we used our patient-derived models to test an immune therapy. And so what this does is it's called a bispecific molecule, and it has one end which grabs onto the T cell and the other end which recognizes here the tumor cell, and here it's using PSMA, and it brings the killer T cell together with the tumor. And we tested that in our mouse model. Again, we had the tumors growing in the mice. And if you look at this lower graph, you can see that the black line is just the tumor growing, and it takes off. And then if you add the T cells without anything to tie them to the tumor cells, it has no effect. But as soon as you put in this drug, this bispecific drug, you see that the tumors shrink and go away. And then if we retreated the tumors, there were 2 mice where the tumors started to regrow, the tumors shrank again. We can see the same thing in the bone. And so the bone is very resistant. It has so many growth pathways and many, many different ways to keep these cells happy, the tumor cells happy, that this is phenomenal. We see the blue line here, again, compared to all these controls. The antibody plus the bispecific plus the T cells completely eradicated the tumor. So that treats, the ROR1 antibody treats ROR1 positive prostate cancer, which seems to be more neuroendocrine type. And this PSMA is targeting more of the adenocarcinoma. So we've got these phenomenal therapies. And one of the things that I told you is that these were the preclinical research done partly in parallel, partly ahead of time to really dovetail and justify and help understand help the clinical trials for these products. So as Dr. McKay mentioned, we have a Simtuzumab plus docetaxel trial for metastatic prostate cancer that is about to, we're just trying to get that one off the ground. We're all very excited. Everything's ready to go. There's just a couple more things to go back on. That bispecific that I was just telling you about that drags the T cell over to the tumor so that it can kill the tumor cells, that is now in a clinical trial that Dr. McKay is the site PI for. And then we have 2 other clinical trials that I didn't mention. And one of them is this PROSPAC active surveillance trial, which Dr. Parsons is the PI of, that we're very, very excited about. I just wanted to quickly say that we're also developing these in vitro models so that we can, they're 3D cell culture models. They're basically mini tumors that we can generate in 3 dimensions, but in a culture dish, straight from patient samples. So we take the patient sample, we dissociate the cells, and we suspend them in kind of like a Jell-O mold with all the factors that they could ever want, and then they grow into these mini tumors. And using these, we've established beautiful mini tumors that look just like the tumors in vivo. And we've tested different things, and we became very interested in something where in our analysis of it, we found that the anti-androgens in these organoids were actually turning down two of the COVID-19 or coronavirus or SARS-CoV-2 proteins that are required for the virus to enter. And so when the virus binds, so this is, not prostate cancer, but we're using these in vitro models now to look, not just to be able to test the prostate cancer therapy, but also as a way to look at therapies for unexpectedly for SARS-CoV-2. And the reason we can do this is that the SARS virus, so we're not using virus, this is all just with looking at the proteins themselves that bind. So it binds with one of those little spike proteins to its receptor on cells called ACE2, but it requires a cleavage. And that's done by this protein TMPRSS2, which is very famous in prostate cancer. It's part of a gene fusion that's in 50 to at least 50% of prostate cancer patients, and it's regulated by androgen. And so what we found is that, in summary, is that we can test therapies not only for the entry for targeting prostate cancer using these mini tumors that we can grow from the people in my lab are amazing. We can take these tissues, like the smallest amount of tissue that you can even imagine, and grow these organoids to test for therapies. We can also use these to test for SARS-CoV-2 therapies because these organoids, these three-dimensional, are much more representative of how the cells respond in us. They actually give much more representative responses. And so using these patient samples we are so grateful to have donated from the patients, we can study all these different therapies, and hopefully, we'll be, we've already started moving them into the clinic because of these tremendous collaborations with Dr. Rana McKay and Dr. Kelly Parsons and Chris Kane and a whole huge team. And just before COVID, this was all of us at a meeting in 2019, and hopefully we'll be doing this again in 2021. So thank you very much.
Thank you, Dr. Jamieson, for that fantastic overview of work going on in the labs. I mean, really exciting to hear today everything from precision medicine, genomics for patients, and then hearing about the cancer stem cell side of things and how research in the lab is really pushing the boundary to move these fields forward. So I will shift gears a little bit here because we're very honored today to have another special guest with us. This is Mr. Lenny Green, who's a patient of both Dr. McKay and Dr. Parsons, and he has an incredible story to share with us. We're so excited to be able to hear a patient's perspective here today, along with the exciting advances in prostate cancer research and care. And I'd like to turn it over to Dr. McKay again for a brief minute just to tell us a little bit more about Mr. Green before he shares his story with us.
I had the privilege to actually meet Mr. Green through the multi-D prostate cancer clinic. And actually he was found to have a diagnosis of prostate cancer and did a lot of research and got plugged in to see my colleague and Dr. Parsons, who you have all met, who's just amazingly wonderful. And when he saw Dr. Parsons was like, we need to get Mr. Green into multi-D prostate cancer clinic for well-rounded, kind of thoughtful review of all of his imaging, all of his clinical parameters, genomics to really optimize, his care in the most thoughtful and personalized manner. And so we met Mr. Green in multi-D clinic and developed a personalized treatment plan for him moving forward. And it's been just exciting to see just how amazing he's done and just his attitude and perseverance and resilience, and exciting to introduce him here to share his story with you because it really is very moving. Thank you so much, and it's such a pleasure to be taking care of you.
Thank you very much, Dr. McKay. It's really an honor to be here and to help contribute to the overall science behind prostate cancer. I'm committed to do whatever I can now and in the future to help other men who may be facing what I'm facing. Early in February, I met with Dr. Parsons, who reconfirmed a previous diagnosis of prostate cancer. I was lucky to bring my case to UCSD because I wanted to go where I believe the best care would be. And after meeting with Dr. Parsons, he did suggest that the multi-D approach would be probably the most helpful. So a month after I first met with Dr. Parsons, we got together with Dr. McKay and Dr. Sandhu, our radiation oncologist, to talk about the case and plan to move forward with a plan which would begin with surgery, followed by radiation, and then medicine supplied by Dr. McKay. And that was the plan. And as you say, sometimes all the best laid plans sometimes are impacted. The beauty of the multi-D approach was that Dr. McKay looked at my case holistically as she gained knowledge of me and my case and began to look at the scans and really just to get an understanding about my disease. In doing so, she identified a small spot on my lower left lung, and just in the spirit of being completely thorough, she decided, look, we need to find out what this is. It could be nothing or it could be something else, but we need to find out what this is before we really move forward. So it began to get some biopsies, additional scans, and ultimately what turned out in mid-March was that the spot in my lung was another primary cancer, adenocarcinoma. And so that immediately stopped some of the progress on the prostate because we really needed to focus on what was taking place in the lung. But the one thing I want to point out is the multi-D approach really allowed us to begin the fight with the prostate by starting the ADT therapy to begin the fight while I was dealing with the lung situation. So in April, late April this year, I had a lobectomy of my lower left lobe, which was, obviously very traumatic in its own right. And oh, by the way, COVID was taking place. But the good news is that the tumor that was removed from me had not spread, and it allowed me to continue on and focus on the fight with the prostate disease. I do want to say that throughout my dealings with UCSD personnel, technicians, physicians, anesthesiologists, I could not have been and can't be more pleased with the care, the approach, the professionalism. I feel like everywhere I've gone, I've made friends, and because people genuinely care, and it's been great. The prostate cancer surgery then was scheduled for the end of June, which we went ahead and did. At the same time, the ADT therapy picked up with another medicine, so we were really hitting it from two sides, the ADT and the surgery. So the surgery took place again the end of June. That proved to be successful as it could be, with Dr. Parsons really doing a great job and providing the best care he could. That healing continued all the way through late October, and I began radiation therapy November 5th, and I'm still in it. I've got about 6 treatments left, but I feel great. I've looked at this entire situation as one that if I can be in the best shape I can be, both mentally and physically, I'll be fit to fight. And really one of the things that's helped me get through this too is really focusing on gratitude and really being thankful for all my caregivers, all the people that are involved in my care. And I'm just so grateful again to be here today, but also to say that my experience at UCSD has been nothing short of excellent. And I look forward to continuing my treatment and praying for a great outcome. But I just want to live up to the expectations of my providers. And I think that's the most important thing. I feel that this is a mission that I'm on, but I don't want to let them down. And so constantly focusing on what I can do to please them and keeping fit and eating right and doing the things that I need to do to be in the best condition to fight. And so needless to say, I'm glad 2020 is going to be over. And I'm looking forward to 2021 in terms of making progress, additional progress on my disease. But I am committed from this point forward to contribute to prostate cancer research, helping others understand it, and just trying to treat people how I would want to be treated in this case. And I know I speak for many cancer patients that are served by UCSD in expressing their extreme gratitude for the work that goes on behind the scenes to help make life easier for those of us that have been diagnosed, but also to help prevent it from those that could possibly get it in the future. So thank you very much for having me.
Thank you so much for sharing your story. And our goal is to cure you of not just one cancer, but 2 cancers. And we're on the path to do that. So one day at a time.
Thank you.
Yeah.
Thank you so much, Lenny, for sharing your story with us. I mean, really what an inspiration, and, the reminder to focus on gratitude and keeping up the fight is so important, at the end of the year and especially this year. So, we really, your story is an inspiration to all of us, and especially myself being a researcher in the lab like Dr. Jamieson is. We don't get the opportunity to work with patients daily like Dr. McKay and Dr. Parsons do. And so to have these opportunities where we bring together scientists and clinicians and patients, it really, really reminds me of the importance of the work we're doing. And, I think puts all the pieces together for us all to see that the progress that's being made and how much it means to patients. And, we're really honored to be part of your care and your progress. So with that, I'll shift over to fielding some Q&A. So I'll go ahead and start. There was a question here just about neuroendocrine variation of prostate cancer. So Dr. Parsons, did you want to comment a little bit on what that variation is and how that affects bone cancer and bone marrow and PSA and prostate cancer?
The short answer is that it's an aggressive version it's actually well said right there. It's an aggressive version of regular prostate cancer. And because it's aggressive, it sometimes requires additional treatments. And yes, interestingly enough, it tends to occur when PSA levels aren't very high. So that is to say, for folks who aren't as familiar with prostate cancer as a lot of other folks on the call, PSA is a blood test for prostate cancer. The higher the PSA is, the more, in general, the more aggressive a prostate cancer is. But paradoxically, in some instances, some folks with aggressive prostate cancers have very low, even normal levels of PSA. And the theory is that these cancer cells are so unlike normal cells in the body that they're not making the PSA protein. And they sometimes require additional treatments, and they can sometimes spread into unusual places. And that's where I'll hand it over to Dr. McKay to pick up with that.
Yeah, absolutely. I think we're actually learning a lot more about advanced prostate cancer. As I stated, one of the only reasons that we know about this emergence of this neuroendocrine phenotype is because we're actually biopsying patients, looking at their cancers under the microscope, and being able to see that, hey, these look different than the primary tumors. And then we're sequencing those tumors and realizing, gosh, the molecular makeup of these tumors are very different. I think historically, we used to think that neuroendocrines were kind of separated from the adenocarcinomas or the regular prostate cancers. But what we're seeing is actually that they can oftentimes co-occur. And some patients may not necessarily have a pure neuroendocrine prostate cancer. Now, the emergence of this is quite low. It's actually just probably about 10 to 15% of patients who have very advanced treatment refractory disease. So it's not a very common phenomena, but it can be one that can be tricky to treat. We actually have several clinical trials that are designed for neuroendocrine prostate cancer. There's a clinical trial of immunotherapy that we're leading here at our institution that's currently open and accruing patients who have neuroendocrine differentiated prostate cancer. We're going to be bringing forth another trial actually in partnership with Duke University and the PCCTC called the CHAMPS trial that incorporates immunotherapy with chemotherapy. And actually that platform trial that I talked to you about, the PREDICT trial for prostate cancer, actually does have a that allows patients with neuroendocrine prostate cancer, which those individuals have historically been excluded from clinical trials, and actually has a treatment arm of a drug called a EZH2 inhibitor, which has shown promise for patients who have a molecular phenotype that is associated with neuroendocrine disease. So it's not a very common thing. I know it scares the bejesus out of patients when they hear the word neuroendocrine, or what can emerge, or if I take this drug, I have the risk of developing that. I think it's not a very common thing that happens. But it is something that we look for. And I think this is why it's important to biopsy along the way and sequence along the way. And, sometimes these traditional diagnostics, though they may be annoying, are actually helpful to better personalizing therapy.
Great, thank you so much, Dr. McKay. We're getting a couple more questions kind of on this topic, if you guys want to kind of give quick answers to those. One is, do neuroendocrine prostate cancer still produce high level of PSMA?
I don't mind fielding that question. It's actually variable. Like I said, neuroendocrines tend to, the neuroendocrine cells tend to not express PSMA, but a lot of times neuroendocrine cancers co-occur with adenocarcinomas that do express PSMA. And so I think that there's actually a lot of what we call heterogeneity, or, I like to always tell my patients the analogy of the gumball machine. And your prostate cancer is like all of these different kinds of clones in there that are all molecularly distinct, and they may all have a different sensitivity to any one given drug. And so there can be co-occurrence of both PSMA adenocarcinoma expressing PSMA, but also neuroendocrine. Obviously, you treat to the more aggressive type, which is the neuroendocrine.
I just wanted to echo Dr. McKay's point about the heterogeneity, and that's why using these models where we get the patient samples gives us much more accurate models for testing therapies. And so we see, that some of the cells have PSMA and some of them have ROR1, for example, in this mixed neuroendocrine adenocarcinoma. Very important.
Great point, Dr. Jamieson. So I missed, did Dr. McKay want to comment on if prostate cancer is hormone dependent?
So initially, most prostate cancers are hormone dependent, and dropping down testosterone levels does result in improved outcomes. But for people with advanced disease, at some point in time, a lot of those cancers do get resistant to therapy. We don't know when that time is going to be. There's certainly an average, but there's some men that can be hormone sensitive for decades, and some men that become hormone resistant pretty early on.
Great. So there were some questions kind of about studies, clinical trials, who's eligible, how can people become clinical patients for trials here at Moores and maybe throughout the state and country. What would you recommend for patients?
Yeah, it's an interesting thread of the questions, and I think it seems to focus on a particular theme, which is how can I get into clinical trials? And I think that we are one center. I think certainly one of the bigger ones in the region. I mean, to be fair, there are other health systems in the region that also offer clinical trials. We offer an entire variety of trials, not just in prostate cancer, but other kinds of cancers that Dr. McKay and I take care of, including kidney cancer and bladder cancer. And they are sponsored both by the government, like the National Cancer Institute and other nonprofit organizations, as well as industry. So folks in the private sector who are developing new medications, and it's exciting to be a part of them, and it's exciting to be able to bring those kinds of clinical trials to patients. Along the lines in terms of UCSD, there was a couple of questions. One coverage, for example, or one question, for example, about coverage, UCSD insurance coverage. I think that in general, it can be flexible in terms of folks coming for clinical trials. For example, we do enroll Kaiser patients and patients from other health system plans, and so those are conversations that you can have, with our group in terms of planning visits. Another question from a gentleman about having seen a few of us and how they become clinical patients or clinical trials patients. Well, the first step is to see one of us in clinic and to ask one of us about it, and to see what sorts of trials you might be eligible for, because not every patient is eligible for every trial. There have to be very specific rules, because the first priority is safety and keeping folks safe when we're evaluating them for trials. Dr. McKay is one of the busiest clinical trialists around. Do you have anything to add, Dr. McKay?
No, I think it's, and some patients may not necessarily need a trial right at that
Yeah.
point in time. And so I think it's all about having a great, good therapeutic relationship with your doc who's taking care of you, to make sure that you're always exploring every single option that could potentially be available when you need it. Sometimes you need it and sometimes you don't. And so it's always just making sure that, you're having those discussions, and always, my strategy is always, obviously we're hopeful and we aim for the best, but always have a plan B, C, D, E in your back pocket with multiple, strategies for tackling how the cancer may, what may happen, because there is some unpredictability to things. I know there's a lot on the chat. I know we got to probably wrap up soon, but I'm so excited. There's a lot of discussion on the chat about PSMA imaging, and that's been a huge breakthrough over the last couple of, I think a week or two ago, when the FDA approved the two PSMA strategies at UCLA and UCSF. We are working with our nuclear medicine team and our radiology department through the chair, Dr. Norbosch, to actually work on bringing that reality here. There's actually a small molecule PSMA that doesn't necessarily require somebody to have a cyclotron on site, that will likely get FDA approved within the next 6 months. And I think this is going to be a reality for our patients here. So we're excited to be able to bring that to you soon.
The only thing I would add to that is I am absolutely just as thrilled as Dr. McKay to bring that technology. I read the New York Times piece and by Gina Colada, who's an outstanding health writer, has been for years there. And there was great enthusiasm, but I always tell folks, it's not necessarily going to be the right way forward for every single patient. And every time a new technology comes out, absolutely, we're all excited about it. We're all excited about bringing it to folks and our patients, but it's not going to be the right fit for everybody.
Thank you so much, Dr. Parsons, Dr. McKay. We'll go ahead and wrap it up now. Just want to thank our exceptional panelists today. Just really wonderful presentations by Dr. McKay, Dr. Parsons, Dr. Jamieson, and of course our special guest, patient Lenny Green. We'll see you soon, and thank you again so much for joining us today.